folks, if you don't mind, we're going to try to get going. And again, apologies that we're starting just a little bit later. The class ahead of us takes a little bit of time to actually leave. They want to have some quiet outside in the area where while they're having their lectures. So I thought we'd start just a little bit later. We'll still try to finish by about 7 o'clock. I'll still stick around to answer any questions that you might have. And again, we have a lot on our plate today. There are a couple of different student groups that want to make some announcements, some really important things that I hope that many of you will take part in, and we'll do that just as we're starting the second hour. Just a quick reminder that you should be thinking about your upcoming term test. There's the breakdown of the term test for you. I'm going to be giving you a mock term test that will be a similar level of difficulty, covering the same topics, and the answer key for you as well. Part of your participation marks actually involves posting an anonymous response. So we're all going to practice the short answer questions together. One of those short answer questions that you practice will actually be very similar to what you actually write on the term test. So my goal is to try to give you as much practice and as much experience in writing the term test as possible. And of course, we'll also be having a review along the way as well. So we sort of have left off looking at the gut microbiome. So I'm going to come back and talk to you a little bit about the gut microbiome again. And today I'm going to be telling you a little bit about Parkinson's disease. Many of you have heard about Parkinson's disease before. Maybe I want to give you a slightly different perspective, an emerging perspective on Parkinson's disease, especially now in relation to the gut microbiome. And it was thought in a very bizarre way up until about 10 years ago when the first reports that the gut microbiome, that something living in your gut that's changed by your diet or lack of exercise, or if you think about it, anything that you ingest, how that actually might affect a neurodegenerative condition was thought to be bizarre, was thought to be science fiction. Like, how could your diet and changing the gut microbiome possibly affect your development of Alzheimer's disease or Parkinson's disease or multiple sclerosis? And yet these papers now in 2017 are thought to be mainstream science. And they're very firmly backed up by a number of research forms of evidence. So I'm going to go through that with you today. I'm going to talk a little bit about food and the impact of food, why food can be considered an addictive substance, and why actually it's really hard, even though you know that you should be changing your diet, and even though you know that it's not great to be eating all of those unhealthy snacks that I provided for all of you, that perhaps it's not as easy to change our diet as we thought. So again, I'll talk a little bit about that, but really I want that to be sort of a minor aspect to it. I want to use that to show that what we ingest actually does impact the gut microbiome, and that's the most important part of today's message. And today I'm going to start the process of getting you thinking about the gut microbiome and Parkinson's disease. So we had talked a little bit about dysbiosis, the dysregulation of the normal responses of the resident bacteria that live in your gut, and that's how we're defining the gut microbiome. And this is what we were looking at. What happens if you disturb that balance? And again, it makes a lot of sense that maybe if you disturb the gut microbiome, the normal resident bacteria, and you enter into dysbiosis, that some other things related to metabolism might occur. That wasn't thought to be all that outrageous, and people didn't really have any major objections to this. So one of the things that started to come out fairly recently in the early 2000s was that when animal models of obesity, and we're going to go back and look at some models of obesity in a moment, that when you look at the end effect, so again, think about this, when we look at all of these different studies today, in the beginning of the studies today, when you look at the end effect, and you have an obese animal, and you take the gut microbiome and you look at what happened to the gut microbiome, perhaps it's not surprising that some of the gut microbiome has changed. They are in a state of dysbiosis. That the dominant gut phyla saw a decrease in the bacteroides and an increase in firmicutes. So that's in animals. And again, you might also look at studies in humans where one of the twins and the other twin is put on a different type of diet. And if you look at these twins on diet A versus diet B, that twins where they have a unhealthy diet, they also show a very similar decrease in the bacteroides, but instead of seeing an increase in firmicutes, at least in this one study, there was an increase in another phyla known as the actinobacteria. Overall, same sort of trend. A decrease in one and an increase in the other. Both of them a decrease in bacteroides. So the shift between these two different phyla, whether we're looking at bacteroides and firmicutes or bacteroides and the actinobacteria, ultimately means that the bacteria are going to be using some of that undigested food, which inevitably hits them when it is found in the colon. And so this means that the bacteria that we have when we are in a state of dysbiosis and in an obese state, the bacteria tend to be able to use that energy from the food in a lot of different ways. And one of the things that also results from this, so that they increase the energy output from the food that is left over when you're in a state of obesity, the other thing that results is when you have a tip in this balance and you start having more of the actinobacteria or the firmicutes, you actually start producing more inflammatory factors. Now, you might have heard of pro-inflammatory factors in every one of your courses. In fact, inflammation is the most common type of universal mechanism in neurodegeneration. In any of the pathologies that you study, inflammation is always a hallmark of disease. And here, in obesity, we are seeing a shift in bacteria and also a shift in the inflammation status. So again, inflammation, obesity, and a change in the gut microbiome were all sort of linked together. And if you're wondering, is this just enough? If I were not to actually have a change in diet, so I'm not actually giving these animals different high-fat foods, high carbohydrate containing foods, if I were to just change the gut microbiome, if I were to implant or transplant the gut microbiome from an unhealthy animal into a perfectly healthy animal, will I see the same changes? And indeed, Turnbaugh in 2 separate papers showed very eloquently, at least in animal studies, that a change in the gut microbiome is sufficient to cause a change in the metabolic status as well as in inducing obesity. So right now, when we're looking at what's happening, we know that there's an obesity epidemic. We know that there are all sorts of problems with being not as well in terms of physical fitness as we would like to be. This has become a real issue for us when we're looking at policy, when we're looking at the overall health status. Inflammation is something that we have to think about in several different instances in this course. So it may surprise you or it may not surprise you that inflammation is one of those markers that we also see in depression. And you might be thinking, like, why do you see a change in inflammatory status when you're depressed? It's not like you're becoming obese. And yet, it's very clear that in certain forms of depression, we see a change in the inflammatory status. So in order to understand like why all of these changes are occurring, a lot of individuals have actually gone back. Is it something in the food? And going back to this neurobiology, are we simply eating more? Is the reasons why we have mental health problems, this rise in neurodegenerative diseases, etc., is related to the gut microbiome? Is it actually related somehow to the food? And individuals in the 1990s started to examine food as a possible reward. We know that there are a number of different things that are used to reinforce positive behavior. Many of you will know about the dopaminergic system. And I'm going to revisit the dopaminergic system for those of you who haven't seen it in a while or need a little bit of a refresher. We know that certain things like seeking food under normal circumstances to survive, whether you are looking for safety or you're looking just to be able to survive, if you need to mate, all of these different things might be related to reinforcing a behavior that is beneficial to us. All of these things, in theory, are beneficial to us, and as a result, in order to reward us for doing something that is good and beneficial to us, we think that this stimulates the reward pathway or the midbrain pathway that we'll see off and on throughout this course. So you also know that drugs that aren't originally designed to interact with the system, artificial drugs, also take over this pathway, this midbrain reward pathway. Many drugs of abuse override this pathway and cause it to signal aberrantly when there really isn't a reward. It isn't something where this midbrain pathway is reinforcing a behavior that's beneficial to us. These addictive drugs, drugs of abuse, generally tend to be very much the opposite. They tend to destroy lives rather than making someone's life better. So again, if you go back into the original literature, when people in the 1960s started to look at different drugs of abuse like cocaine, oftentimes it was really hard to distinguish that this artificial drug like cocaine was actually working in exactly the same ways as normal reinforcing types of stimuli. So people would say, wow, you know, like getting a hit of cocaine, I'm so hungry for my next hit of cocaine. Again, sort of looking at the way in which this midbrain is reinforcing this, or I like the taste of cocaine, or it makes me feel like I'm having sex, it's as good as sex, etc. All of this suggests that these drugs of abuse do indeed stimulate this particular pathway. Now this is a diagram that I want you to know in a little bit of detail, not in all of the detail that's highlighted for you here. Hopefully it's a review for the vast majority of you, so it won't be very challenging for you. But when we're looking at the dopaminergic projections, and we're going to be concentrating on dopamine throughout today's lecture, when we're looking at these dopaminergic pathways, there are actually very few nuclei or sets of neuronal cell bodies that actually produce, synthesize, and then release dopamine. And the vast majority of them are found in these areas, in the midbrain. One of those areas is known as the ventral tegmental area, shown here in red. The cell bodies of the neurons that sit in the ventral tegmental area send their axons to a number of different groups of neurons in the ventral globus pallidus, as well as in the nucleus accumbens. And this is the midbrain system that I've just been explaining to you. We get the release of dopamine, or changes in the amount of dopamine that is signaling in this particular pathway. And again, these mesolimbic pathways that I'm highlighting to you, and I'm not showing you the mesocortical pathways that go all the way into the prefrontal cortex, these are incredibly important. These are the ones that reinforce behavior, that allow you to survive. But they're also the same pathways that allow people to become obese. Food becomes an addictive substance. You like eating, you like the taste of food. Certain foods are things that, again, you don't need just to survive, you actually are seeking out these different food types. How many of you, for example, have ever had a food craving? Okay, so how many of you might be thinking right now about dinner after lecture, and you're already thinking about what you want to eat? So that's that midbrain structure that is signaling, wow, I would really like to go out and grab a burger, or grab some pasta, or a really nice salad. All of these things are signaling through this mesolimbic system. And we're going to come back and look at this in the next slide in a little bit more detail. That's really just an overview, again, a review for the vast majority of you that I really want you to know. And we'll come back and talk about some of the other points. You don't need to worry about them right now, later on in this particular course. So we don't have to worry about any of the structures here in blue. We'll talk about those later. And I'm going to start talking about the reverse pathway a little later in the course as well. I don't need you to memorize everything that's on this particular slide. This is just a different representation. Here, what we're looking at is that nucleus accumbens neuron, and it's shown here as NACC or NAC. That's an abbreviation for the nucleus accumbens. The nucleus accumbens is where we are going to have these different dopaminergic receptors, and these dopaminergic receptors are going to bind to the dopamine that gets released from those different neurons that are coming from the ventral tegmental area. But we know that these different drugs of abuse can act in different places. We know, for example, that opiates, alcohol and nicotine, cause an increase in the release of dopamine, as well as allowing dopamine to stay at this particular synapse for longer periods of time, which means that we stimulate this nucleus accumbens for longer periods of time. This becomes a real issue, not only because we're stimulating dopamine receptors, because that's natural. The unnatural part of this is where dopaminergic neurons are now becoming overly sensitized. It's not just the release of dopamine, it's the activation of these downstream pathways downstream of this dopamine receptor, where we will start to activate cyclic AMP and calcium pathways and MAP kinase pathways that cause this particular neuron that's responding to dopamine, this nucleus accumbens neuron, to now start increasing the expression of genes like PREB, or cyclic response element B, or PREB, and that's how I'll normally abbreviate it. You might start to see the increased expression of delta phos-B, as well as NF-kappa B. Just one question for those of you who are in immunology or taking other courses like NGY. What's the significance of NF-kappa B? And I think we talked about this just a little bit in the last lecture as well. NF-kappa B, have you heard it in other lectures? Do you remember what NF-kappa B does in your other courses? Yes, did you have a... So it could be a transcription factor, and what is it involved in? What transcriptional pathways? Yes, Zoe. So it normally stimulates the cytokine pathway. Cytokines are part of that inflammation process, and it's thought that, again, these long-term changes that occur in the brain in response to having this increased level of dopamine through these different types of drugs might actually be really important. And again, the transcription factors, if you've seen them before, great. If it's new to you, perhaps spend a little time going over it, understanding that this is that nucleus accumbens that's part of the midbrain that is responding to dopamine release. So again, we know that different drugs of abuse will target different parts of this mesolimbic system. So the midbrain system that I just mentioned to you. We know that certain drugs of abuse, things like cocaine, amphetamines, opiates, etc., they can target a number of different places, including the nucleus accumbens, and in some cases the VTA as well. We know that alcohol, barbiturates, and benzodiazepines will tend to preferentially cause the increased release of dopamine to activate the nucleus accumbens cell. So whatever is happening, something is changing the overall levels of dopamine. And this increase in dopamine that we're seeing is actually what we think is the key driving motivational factor that's involved in things like addiction. And again, there are a number of different hypotheses that have been put forward, specifically related to the nature of dopamine. I'm not going to go into all of the different aspects of dopamine as a neurotransmitter, but it's thought that it's, again, a really powerful motivating signal for a number of these different downstream pathways that involve the hypothalamus. So it doesn't end at the nucleus accumbens. Why it's actually subverting the drives for things like survival, and why it's actually linked to things like survival, taste, sex, etc., is because ultimately that nucleus accumbens will also have projections to areas of the brain like the hypothalamus. So it doesn't just end at the nucleus accumbens. Always keep in mind that a circuit is really never a closed circuit, and it has a number of collateral branches. And in the modern neuroscience, we look at circuits now, and one of the other aspects of this mesolimbic circuit that all of you know, you can tell me about the VTA nucleus accumbens, but beyond the nucleus accumbens, it also goes and signals to the areas of the hypothalamus, which we know are important in things like homeostasis. The fact that you eat, drink, etc. is controlled and mediated by projections from the nucleus accumbens as well. And you don't have to worry about this particular diagram. So this became a real issue. And I know it sounds ridiculous, but in the 1990s, so the era that I was growing up in terms of neuroscience, there was this real issue that, obesity and food addiction, those aren't actually real things. Obesity, because you overeat, is not a real thing. It was a psychological construct. You actually lack willpower. And that was what it was thought, that it was impossible because food is intrinsically a natural sort of homeostatic pathway. We need food. Why would food be an addictive substance if it's something that we actually need in order to survive? And this idea that food could be addictive and overeating as a result of stimulating and this mesolimbic pathway, where eating became a very addictive process, was thought to be not a neurobiological phenomenon. It was thought to be completely psychological. Like you just ate because you enjoyed eating. There was no biological mechanism behind this whatsoever. and was thought that it was simply a failure in your willpower to be able to do this. Of course, now, in retrospect, we know that this is probably not true. And again, we won't go into all of the details. You don't need to really worry about this particular slide. And the pleasure that's associated with eating and the release of dopamine, which signals back to the, from the nucleus accumbens, the hypothalamus, and makes a return circuit back, this hedonistic pathway was thought to be really important. That's why dopamine and the release of dopamine after you eat was thought to be really important. And this, again, was really controversial. So are we actually eating too much because it is addictive, and it's really all due to an increase in dopamine release? And can we actually explain all the things that we're going to talk about later, the dysbiosis in the gut microbiome? You eat more because food is addictive. You change the way you eat because food is addictive. Can we relate it all back to this hedonistic pathway? So there's a lot that sort of suggests that this is somewhat true. I know a lot of you are also taking psychology, so you will probably have heard of this theory in psychology as well. So how many of you have ever eaten a chocolate sundae? Some of you are honest enough to admit that you've eaten a chocolate sundae. There's a lot of sugar in a chocolate sundae, but you kind of like the taste of a chocolate sundae, right? You just eat chocolate sundaes for no reason. You like it, that's why you eat it, right? Thank you. Yes. Okay. So most of you would never sit there and in one sitting. You might if you were dared to do this, but none of you would actually sit there and actually eat the equivalent number of sugar packages, right? Again, the answer is supposed to be no. Thank you. No, you probably would. So there's something about the actual way in which you eat that makes this actually really important. There's an element of taste that's involved in this, so even though you're getting the same amount of sugar, the sugar is presented to you in a slightly different way. There's more taste associated with the chocolate, etc. And this stimulation of these various pathways are actually really important in ingestion. And it's thought that this is one of the reasons why we will tend to continue eating even though we're full. And again, this gets into a lot of different psychological constructs, but one of this leads to a theory that was developed called dynamic novelty or dynamic contrast. Have any of you in psychology heard of dynamic novelty or dynamic contrast? No? Not at all? Can you make any guesses before I get to the next slide what dynamic novelty or dynamic contrast is? Any theories? Any ideas? You can say whatever you will. I'm happy to translate for you. Yes? Something new is more interesting to you. So how many of you have the iPhone 7? Oh, good. I'm glad that everyone's being a responsible student, not going out and buying new gadgets. Great. How many of you have the iPhone 6? Okay. How many of you who have the iPhone 6 will purchase or think about purchasing the iPhone 10? Okay, I would too, maybe. And this is because it's new, it looks different. And this is this idea that when something is new and different, and it's also stimulating a number of different pathways, that this actually will increase the likelihood of stimulating the midbrain pathway, because one of the other things that is really important about dopamine is that it's involved in learning, and it's a reward-based learning and prediction pathway. And so when you can't predict something, like, wow, this is new, I've never tried it before, it looks different, it stimulates the release of dopamine, not through the hedonistic pathway, but through this other reward-based pathway. Whatever is releasing dopamine will cause it to potentially be somewhat addictive. And again, this has actually been really important. People spend a lot of time and money trying to figure out how to get you to eat their food, right? So the fact that some of you ate my Oreos, I won't say who, actually, Oreos, there was a lot of research that went into Oreos. Because if you think about how addictive Oreo cookies are to some individuals, there's a lot of science. So it uses dynamic contrast, dynamic novelty, right? So there's this sort of slightly bitter tasting chocolate wafer, and then there's a sweet, creamy filling, and it's crunchy. So when you stimulate all of the different senses in terms of taste, in terms of being able to hear the sound of the crunchiness, all of that was actually well thought out. in terms of this dynamic novelty and contrast. And here's a good example of this dynamic contrast. Drunowski was the very first individual to start thinking about this. And one of the things that he looked at was a group of mice. He looked at a group of rats. You can go back and check out his original article back in 1992. It was completely laughed at the time that it was put out there because no one actually thought that this was going to be correct. But one of the things that he did was he took a bunch of rats and he started to do some experiments in terms of the amount of sugar that they were allowed to eat, and he checked the amount of fat. That's of interest to us now because all of our foods contain lots of sugar and lots of fat. And one of the things that he started to do was he wanted to know, like, if I give all these rats exactly the same amount of sugar, can I get them to eat more sugar? And once they reached a certain level of eating sugar, they actually didn't really want to eat any more sugar. But in order for them to eat more sugar, one of the things that he found was if I mix a little bit of fat in with the sugar, they'll actually go and eat more of that sugar. And so one of the things that he found was the fattier the mixture was, so if you had skim milk, which has very little fat, or you had very thick cream, the animals that were exposed to having sugar and cream, they actually ingested much more sugar and cream. So think a little bit about what you order at Tim Hortons. Your most popular drink is what? The double-double, right? So at one point in time, I used to drink the medium-sized double-double, but over the course of about a year and a half, I went from drinking a medium-sized double-double to an extra-large quadruple. Because I love, to be honest, I love the cream, I love the sugar that was in there. It became, for me, like really addictive. So this importance of having both fat and sugar, so it's not just an increase in sweetness. The sweetness itself, like you'll stop after a certain point. It's just too sweet, I won't eat it. But if you put some fat in there, it actually becomes more addictive. It stimulates more dopamine release. It stimulates more of that mesolimbic pathway. And this idea that you have these contrasting tastes that these rats are not exposed to, and having this dynamic novelty and this contrast. So having both the novelty contrast was not just having sugar, but having fat with sugar. That was the key point in turning their food intake behaviors to becoming addictive type of behaviors and changing their preferences for food as a result of this. And think back again to your own particular food taste. Some of you like having sweet and salty. Some of you will like having bitter tastes that are mixed with others, sweet tastes. So this is actually a big part of the entire food industry. So again, this other experiment is one that I would like you to make sure that you know, because it also highlights in the same way the experiments that Drunowski had shown earlier. So there's this really interesting phenomenon, and we don't really know how to explain it in biology yet, so someone really bright will figure this out. What do you guys know about lab animals? Do you know anything about lab animals? Nothing. I'm hearing they're cute. Okay, yes, cute. Let's get past the cute. Yes. Okay, so they're restricted in their movement. What else do you know about lab animals? Are they genetically identical, or they're all over the place? If I order them and I want to do experiments on them, are they genetically all the same so I can make sure that my cohort is the same, or are they like all different genetic backgrounds? Yes, Andy? Okay, I could order different strains, but in those different strains, are they genetically identical or genetically different from each other? So most of them, yes. So they could, between strains, they are genetically different, but in one strain, when I do experiments on mice and rats, I want them to be consistent, and so I make them all genetically identical. But here's this weird thing. All of them are genetically identical to each other. They're kind of like having twins, or like multiple versions of yourself. They're not completely identical, but they're so close to being identical. So when you take a look at these different animals, there's this really interesting phenomenon. And we're going to come back and see this exact same phenomenon with depression as well. So we'll see this The proportions are a little bit different, but the same idea holds for depression. Sprague Dawley rats are these white rats. They're inbred rats, and they're genetically identical rats. And if I take 24 of these Sprague Dawley rats, and I give them all the same diet, and this diet happens to be a high-fat diet, one of the most interesting things that happens in biology is that these genetically identical rats, all 24 of them, given the same diet, they actually separate themselves out into two different groups. One group becomes what is known as a diet-induced obese group. It makes sense. They've never encountered high-fat foods before, but now that they're getting high-fat foods, they just keep eating. They eat much more than they normally do, and as a result, they become obese. That makes sense. Makes perfect sense. And that's one group. But out of that 24, even though they're exposed to the same food, there's also an obesity-resistant group. This OR group is one that, even though they're genetically identical, and even though they're exposed to the same food, they actually won't eat much more food than they normally would. They do not become obese. Can you explain this? Good. Don't try to explain this. So we don't understand this phenomenon quite yet. They're genetically identical. They come from the same background in terms of genetics, but one gains weight and becomes obese, and the other one doesn't. And wow, great story, end of story. But again, Drewnowski's not satisfied with this. So high fat, he already knows that, for example, high sugar, animals will stop eating. So going back, he does the reverse experiment. Instead of adding fat to sugar, how about if we add sugar to fat? And one of the interesting things that was found, after this 2-week protocol, after we get this really clear separation where we have a group that is obesity resistant, and we have a diet-induced obese group, and they're very clear, we see a change in weight, and the other group doesn't change in weight. So when he goes back and he actually starts to look at what happens if I start introducing high sugar. So again, it makes perfect sense that the diet-induced obese group actually continues to gain even more weight. But when he gives high-fat and high-sugar to both groups, the obesity-resistant group, the one that normally didn't want to eat anymore, now with this new fat and sugar together, they actually start eating more than they were before. They also become obese. And this is one of the reasons why Drewnowski and others were saying that the different ways in which we present food are changing our eating habits. And by changing our eating habits, because we are now ingesting things that have these combinations much more readily, and they're much more easily attained, that we're changing our eating habits, and maybe we're changing our gut microbiome. And again, this is that idea that the different groups that come up with the different foods that are introducing dynamic novelty. They're introducing both high fat and high sugar, stimulating your mesolimbic pathway a lot, and it's at work in changing your overall feeding behavior, not just in animals, but also in humans. So again, I'm not going to go into a lot of detail right now, but you can actually go back and test. So it's one thing to say that, okay, I get the fact that dynamic novelty, changing like what the animals normally eat, having a number of different sensory pathways being activated, the fat and the sugar pathway, may be causing them to eat more, but is it truly addictive? Like that's one question that isn't answered in these studies. So again, going back to classical behavior, do you remember in your psychology classes what a progressive rewards model system is? What is that, or what could it look like? Any ideas? Progressive rewards. No idea. Nothing. No guesses? Yes? More rewards? The farther you go. Okay, so that was a really good explanation. I love that explanation. I'm going to translate that explanation and make it sound more neuroscience-y. Is that okay? Okay, awesome. But thank you for doing that. So, as your colleague pointed out, one of the things that happens in progressive rewards, for you to get one reward, you have to do a certain task, whatever it is. It might be pulling a lever, it might be pushing a button, and a rat will do that. And it will get a reward, and we'll understand that it gets that reward, and that's the end of the story. A progressive reward model is a little different. The rat has to understand that in order to get a reward that it feels is rewarding, it has to continue to ramp things up. So it's not one button push that will get it the next reward. It has to actually push the button 3 times, or they have to push or pull a lever 3 times to get the next reward. And the next one might be 10 lever pulls before the rat gets its reward. And we want to know, how far would a rat or a mouse be willing to do these progressive rewards? Will it stop at 20 lever pulls before it gives up? It just becomes unsatisfying. And is food also equally addictive? Because one of the things that we normally do is to use these types of studies in addiction studies where an animal will receive a drug of abuse reward. When they will pull a lever, they'll get a little shock that stimulates the midbrain. So how many of these things will they be able to do before they reach the breaking point where they stop? I don't really care anymore. It's not worth my time and effort to put any more work into this. It's kind of like thinking about your GPA. At what point do you sort of give up on things and kind of just say, that's enough. I can't do much more than this. So when you look at these compared to like stimulating the mesolimbic pathway directly, injecting in like cocaine into this pathway, how far along is a food reward in this particular pathway? So sugar and fat combined are just below cocaine in terms of where the animal will stop. So is it addictive? There's a lot of evidence to suggest that foods can be addictive in animal studies and in human studies. So there's a lot that we need to sort of think about. So is food an addiction? I would argue very strongly yes. If you wanted to stop an addiction, and again, it's not in your notes, but maybe you can tell me a little bit about your neuroscience backgrounds. How do you actually stop an addictive substance from working? Like, you can give a drug. Do any of you know what that drug is? Some of you might be in farm talks. Yeah. You could give naloxone, right? So it's an opiate receptor blocker. And if you were to give an opiate receptor blocker, and you don't need to write this down because it's not in your notes, many of these different forms of addiction can actually be overcome. And it's the same thing for food addiction. When Bernowski and others look at giving naloxone and then seeing if they'll continue to eat these addictive foods, they stop doing so in the presence of naloxone or naltrexone, which is another opiate receptor blocker. Okay, so that sort of takes us to going back to look at why we're looking at all of this. Ultimately, we eat these different foods that are high in fat, high in sugar. We know that these could potentially be addictive. We're not able to control what we intake. This might be changing our gut microbiomes, and by changing our gut microbiomes, we may be heavily influencing our health status, even though we may not have been aware of it. Just out of curiosity, because we have a couple of minutes, and again, this is just an open question. What else do you guys eat? So you eat food. What else do you eat? It's not a trick question. You have to think a little bit, but you don't have to think too hard, hopefully. Yes?
Like health food products.
Okay, health food products are another type of food, but that's great. Yes?
Unnatural chemicals.
Unnatural chemicals. Could you... Cool Whip, okay. I'd still consider Cool Whip, although it's an unnatural chemical, to be a food product. Great. Nice. Yes?
Water.
Okay, water. Definitely we drink water. Yes?
The air.
Air. Okay. Kind of like it. Nice. Yes, in the middle. Medicine, drugs. Okay, so let me, again, most of you probably don't take any more medicine than an occasional Tylenol or Advil. Am I right? I'm hoping. You might eat vitamins. Do any of you eat vitamins? A couple of you, okay. How many of you have parents who are somewhere between 50 and 60 years of age? A lot of you, okay. So when you get older, one of the things that happens is you tend to eat a lot more drugs. So you take medicines, and these medicines actually have really good compounds that will help in trying to fix or maintain different things that are going wrong in your parents. But what else are you ingesting when you take in medicine? And this is actually a really big thing right now, and we're rethinking this in a lot of ways. What else do you find in medicine? Those of you who are in farm talks, for example. Yes?
Sugar.
Sugar? Could have some sugar in there, definitely. Sometimes medicines are flavored so that they taste better. Sure. What else? When you think medicine, what do you normally think of? Yes? Antibiotics, okay? Possibly antibiotics, and we'll talk about antibiotics later on. Yes, William? Gelatin, gel capsules, sure. But when I'm taking Advil, or I'm taking like a hard pill, what else do you sort of notice about it? So nowadays, you just go to the pharmacist and you get these little pills, right? In the old days, you used to get this powder, and you had to do something with that powder. But in order for you to maintain the shape of that pill, it actually contains a substance known as a binder. Have any of you heard of these binders? These binders that allow a pill to change, to keep its shape, that makes it a circle or make it an oval, are actually impacting our gut microbiome. We never knew that until fairly recently, and these pills that are now being ingested are actually changing our gut microbiome as well, and you might be interested in following up on that. But I'm going to be talking to you about a slightly different model. Now, I'm introducing this for a variety of reasons. So hopefully, we're at the stage where we understand that there are lots of things that are happening to our food, an increase in dynamic contrast and novelty, like having two different things, high fat and high sugar, allowing us to eat more, stimulate our mesolimbic pathway, become addicted to certain foods that change perhaps our gut microbiome status. Well, one of the things that we are more and more sure of, and there are more and more papers, that somehow this gut microbiome is actually being impacted by the changes in our diet that we're talking about here today. And one of these papers that came out in Cell, and Cell is a very reputable paper. It is one of the highest cited papers that we have in science, and it's the paper that if you were to ask any researcher here at U of T, what papers would you want, or what journals would you want to publish in? Nature, Science, and Cell are always at the top. It's one of the highest ranked papers, and this journal had come out with last December the fact that the gut microbiome actually regulates motor deficits and neuroinflammation. Again, inflammation is going to be a big part in this course, at least in an animal model of Parkinson's disease and carrying over into the genetics of Parkinson's disease. It was, again, one of those really interesting papers that a lot of people kind of sat there thinking a little bit about. The other reason that I'm putting this out here is related to this. And this is sort of the assignment that I want you to be thinking of. In most journals now, they not only have an abstract, which was, again, very common in my era, a small summary of the major findings in the paper. But nowadays, most journals are also including what is known as a visual summary, a visual abstract, a graphical summary, whatever you want to think of. And this basically tells the entire story of the paper in about 3 or 4 panels. So here's panel A, panel B, and panel C. And if you were to follow this, the story basically tells you exactly what they found in this paper, but not going into all of the different details. So we know, for example, hopefully after the last lecture, that we normally have a gut microbiome, and this gut microbiome will produce, as we'll see a little bit later on, short chain fatty acids that will actually change the status of these different microglia in the brain, and this can also impact the brain directly in terms of what happens to the normal physiology. We know that if we were to get rid of some of these gut microbiomes, some of the bacteria, that in fact we don't have this short chain signaling, we don't get activation of microglia, and actually reduces the pathophysiology within the brain. We don't see the motor deficits. And again, within these 3 panels, it actually shows you all of these different things. And the last thing that they do in this paper is to look at, if I replace the gut microbiome with something that comes from a Parkinson's disease patient, it will actually cause the enhancement of motor dysfunction. They look like they have Parkinson's disease. The animal models look like they have Parkinson's disease. That is the entire paper summed up for you. And I'm going to take you through that paper and give you a little bit of background on Parkinson's disease. But this is what I want you to be thinking of when you're looking for a paper. Could I make that same sort of summary? It's not as trivial as we think. And again, I'll be working with you on that, and Darren and others can also help you with this process as well. Do you have any questions on this before we go on? Any questions? No? Okay, I'm probably going to take you through a couple of slides, then we're going to try to take a quick movement break, then we'll take our regular break, and then we'll finish off today's lecture. So we know that neurological dysfunction is actually the basis for a number of different human diseases, particularly things like neurodegeneration. It also turns out that things like psychiatric and other developmental conditions actually have a number of hallmark features. One of the things that, again, may be surprising to you, or maybe not, if you're taking LMP courses, for example, you'll have heard of this. Most neurodegenerative conditions, and even some neurological conditions, and even some psychiatric conditions are often accompanied by the preponderance and overexpression and overabundance of misfolded proteins. There's always something. So Huntington's disease, for example, there's an accumulation of Huntington proteins. If we're looking at Alzheimer's disease, many of you will have heard of beta amyloid or about tau. And when we're looking at things like Parkinson's disease, we also have misfolded proteins that are accumulating within certain groups of neurons. And when we're looking at this now, in 2017, this is actually really, really important. Something causes this misfolding. We don't really know all of the details right now, but we can identify when this process is occurring, and that this is a hallmark feature of these different neurodegenerative conditions, whether it's beta amyloid in Alzheimer's, whether it's the Huntington protein being overexpressed in Huntington's disease, whether it's alpha-synuclein in Parkinson's disease. All of them are misfolded, they're somehow causing damage to neurons, and this becomes a big issue. So Parkinson's disease is actually one of the most common, it's the second most common neurodegenerative disease in the United States. It affects an estimated 1 million individuals. So in Canada, because we scale things back by an order of 10, probably about 100,000 individuals in Canada have Parkinson's disease. It's one of those really important features that it is a movement disorder. You don't move like you normally do, and that's the characteristic feature of Parkinson's. There's a motor deficit. You just don't move like you normally would. It includes having resting tremors, so if you see someone sort of sitting there and they have their hand kind of shaking, that resting tremor is characteristic of Parkinson's. They also have this muscle rigidity, and this muscle rigidity actually comes into play after the break, and we'll talk about that in a moment. Bradykinesia, what is that term? Bradykinesia? Yes? Yeah, the slowness of movement. All of the movements that you have are much, much slower than they normally would be. So 100% correct. And there's always problems with walking or an impaired gait that's associated with it. The problem with everything that we talk to you about in terms of neurobiology, especially of neurodegenerative conditions, which all of you are fascinated by for a variety of reasons, is the vast majority of this is really unsatisfactory. So as neurobiologists, all of you should actually be outraged that we don't know what causes Parkinson's disease. We only understand 10% of Parkinson's disease. 90% we have no idea. Like, you've got Parkinson's disease? I'm sorry, I have no idea why you've got it. Could be environmental, could be genetics and environmental, I have no idea. Isn't that disturbing to you? That even though this is the second most common neurodegenerative condition in the United States, we don't understand how it occurs. We know that there's misfolded proteins, but the vast majority, 90%, is not due to genetics. We don't have any genetic background issues, and yet you develop Parkinson's disease. You don't seem outraged. You should be outraged, because neuroscience is supposed to give you these answers, and we haven't. And we haven't done this for a very, very long period of time. We also know that in these neurodegenerative conditions, we have a misfolded protein. The characteristic misfolded protein in Parkinson's is this protein here known as alpha-synuclein, often abbreviated here as alpha-syn. And this is sort of thought to be pathogenic. In the 10% we do understand, there's a problem with alpha-synuclein, so that makes perfect sense. But the 90% of the other forms of Parkinson's, where people have problems moving, they have problems being able to walk properly, the other 90%, there is no genetic predisposition to have alpha-synuclein misfolding, and yet it still happens, and we don't understand why. And this, again, to me, is something that is completely unsatisfactory. I'm hoping it's unsatisfactory for some of you, because you might be really interested in trying to tackle this and trying to do some research to try to figure out why we don't understand all of this. We know that these different synucleopathies, that includes Parkinson's, also includes other parts of the brain that are involved in multiple system atrophy, also involving substantia nigra and other structures. It can also involve Lewy body disease, etc. And one of the most surprising things is that maybe these misfolded proteins, under the right conditions, can actually spread from one brain to another brain. And again, if we have time, we'll come back and talk about this. So I think now, because it looks like your energy level is a little bit low, I think now is actually a pretty good time to do a movement break. It will only take 3 minutes, and then we'll still have our full 10-minute break after. So just bear with me. If you're feeling particularly self-conscious, look at me. Okay, I'm no longer breathing in, and I'm just letting it all go. So feel free to let it go as well, and don't feel self-conscious. If you want to join me up on stage, you're more than welcome to do so as well. And I think I'm going to invite Victoria and some other people up here as well. We're basically going to be following along with the particular video that's shown up here. If you can stick around, that'd be great. It won't take long. Try not to hit the person beside you. You don't have to stand up if you don't want to. You can do most of this sort of sitting down for this particular exercise as well. So are you coming up here, Victoria? It's actually not so bad. This one I think I can actually do. Yeah, whenever you guys want to get rolling, you're starting too soon. Okay, let's hold time. And can you hear it on the speakers or not? Okay. Okay. Probably do it directly off the
and 1. Tilt your head towards your right shoulder and hold. Slowly transition to the other side and hold. Slowly come back to neutral position. We're going to take your right arm across. Bring your other hand to your elbow and pull back and hold. Slowly come out of the stretch. We're going to repeat on the opposite side. Take your left hand across, bring your right hand to your elbow and push back. And slowly release the stretch. Great job. We're going to bring your arms forward. You're going to push your hands towards the front. So you bring your head down and hold. your head up. Bring your hands back. And look up. Bring your hands forward. And release your arms. Great job. We're going to end off taking 2 deep breaths in. Inhale. And release. One more time. Take a deep breath in. Give it all you got. And slowly release. Thank you.
folks, we're going to get going in 1 minute. Actually, there are 2 student groups that have some really important announcements for you for some important events that are occurring Thursday. So if you guys want to come up, I think there's a NOS event and there's also a Scientista event as well. So you can introduce yourselves, tell us a little bit about when, what's going to happen, why people should be coming out to do this.
Hi everyone, my name is Amy and I'm the Vice President of Academic Affairs for the Neuroscience Association for Undergraduate Students. So we're the Neuroscience Council that represents anyone who's doing a major or a specialist in neuroscience. We run a lot of cool events throughout the year. We have a conference coming up on November 4th that's on mood disorders. We also run a life sciences competition in March. So it's a lot of fun. I would highly recommend coming out to our events. And we also have an event tomorrow that's basically pizza with profs. So you meet all the neuroscience profs and HMB profs. And you also there's also an opportunity to run for 2nd or 3rd year rep. So if you're interested in joining the council, I would recommend coming out. Or if you just want to vote. Thank you.
Hi everyone, I'm Laura and this is Clara, and we're from the Scientista Foundation here at U of T. And we're happy to announce that we're also having an event tomorrow night. It's our first academic event of the semester.
Ours starts an hour later. Yeah, so ours starts at 5, theirs starts at 4. And we're having a guest lecture by Dr. Rosanna Olson. She's a renowned neuroscientist here at U of T, and she'll be giving a talk about her work, and there will also be a chance for you to ask her some questions and get to know some people from Scientista.
And there will be snacks.
Snacks.
Okay, so before we get back into the gut microbiome, let me tell you about alpha-synuclein. It's going to be really important as an animal model. So generally speaking, when we look at a misfolded protein and we use it to identify a neurodegenerative condition, there's always a hallmark protein. In our case, in Parkinson's disease, and many of those alpha-synucleinopathies, so things like multiple system atrophy, or we're looking at other forms of degeneration that involve dopaminergic neurons most often, we're looking at alpha-synuclein. We're also going to see alpha-synuclein in a transgenic animal in a few moments as well. So alpha-synuclein is actually a normally occurring protein, as you would likely guess. It exists as these little monomers, and these monomers actually begin an aberrant form of accumulation. It's a stepwise process. It goes from these monomers to these protofibrils and then eventually into these fibrils. Regardless of how these different oligomers form, they will eventually become very, very insoluble. They become this almost like precipitate that is existing within the cell. It's very deleterious to the cell. It injures the cell, and eventually these cells start to accumulate so much of these fibrillar proteins, which in many ways look a lot like beta amyloid deposits. These are again in dopaminergic neurons though, that you actually will get the generation of what is known as a Lewy body. And there are different forms of diseases like Lewy body dementias that have alpha-synuclein accumulating, forming Lewy bodies, and then causing dementia. So these misfolded proteins that begin with alpha-synuclein are actually a very big deal for us. It's one of those hallmark features that we can actually look at. And in a damaged group of dopaminergic neurons, and dopaminergic neurons we talked about in terms of addiction and others, we know that dopamine is also really important within the substantia nigra for regulating the smoothness of movement. And in the substantia nigra, as an individual starts to develop Parkinson's disease, we get more and more of the accumulation of this alpha-synuclein. Again, just as I told you before, you should be looking at this through the lens of being young. You have a lot of different ideas, and you have a lot of different perspectives on things, and we don't have all the answers. Just as I told you that even though we understand that for most diagnosed cases of Parkinson's disease, on autopsy and pathology, we will be able to find alpha-synuclein deposits that likely result in these Lewy body formations within dopaminergic neurons in the substantia nigra. We don't really have a good cure for this either. We can treat this for a couple of years, maybe a little bit longer. We can use different types of drugs, but these drugs like L-DOPA therapy often lose their effectiveness over a very short time course. And even if we're giving dopamine agonists or dopamine receptor agonists like bromocriptine, because we're losing dopamine and there's no way for us to get these neurons back, ultimately this is not a win situation. So we don't have an effective cure. We can't replace these dying dopaminergic neurons in the substantia nigra, and as a result it becomes a really big issue for us. Again, the vast majority, 90% of these cases, are actually sporadic. We don't know what is causing these disorders. We know that they're non-genetic. We can only account for about 10% of the cases. We have 2 mutations in one of the ubiquitin ligases, the E3 ligase, as well as Parkin ligase, as well as in the alpha-synuclein, but the vast majority we don't know. We don't know what causes Parkinson's disease. 90% of these are sporadic. Again, we don't have a lot that we can actually tell you about Parkinson's disease in general, other than we know that dopaminergic neurons in an area of the brain known as the substantia nigra are going to be dying. And we know that even as you age, that you're going to be losing somewhere between 1 to 5% of these neurons as you age normally per a set time period. So over the course of 2 to 5 years, you might lose 2 to 5% of these neurons. The longer you live, in other words, the more likely you're going to be losing these substantia nigra neurons. But when you have the alpha-synuclein depositing, that is sped up tremendously. You are losing much more than sort of the 1 to 2% that you might normally be losing for every 2 to 5 years. The other thing that makes this really interesting is that within the substantia nigra, the dopaminergic neurons are often associated with neuromelanin, which is a pigmented form of a protein. And dopamine and neuromelanin, again, are very highly concentrated within the substantia nigra that is being lost in Parkinson's for reasons that we simply don't understand. The typical onset for Parkinson's can be in middle age, usually 50 years of age and beyond. But again, we don't really understand why some individuals with no genetic background, nothing wrong with alpha-synuclein, no mutations in alpha-synuclein, no overexpression of alpha-synuclein, nothing that would suggest alpha-synuclein is going to go wrong, they develop Parkinson's disease as well. And these individuals that have a genetic predisposition often develop Parkinson's disease much earlier, under the age of 40. So again, this is just an overview for you. If you're looking at the midbrain, so again, this is not the same midbrain group of neurons that I showed you with the nucleus accumbens or the VTA to nucleus accumbens, but it's very nearby. The substantia nigra and VTA anatomically are actually very close together. And again, when you learn about that in your neuroanatomy classes, you can ask your neuroanatomy prof, do both groups of neurons, because both the VTA, do you remember what the neurotransmitter was within the VTA? What does it produce and release? Dopamine. And very nearby, we have another group of these neurons, these dopaminergic neurons in the substantia nigra. You can ask your prof whether or not the VTA neurons in Parkinson's also degenerate as well. And it's a very interesting story that we don't really have time to get into. We know that many other neurons, it's not just the substantia nigra. If you were to look at the Parkinson's brain, many neurons, not just the dopaminergic substantia nigra neurons, are lost. And we don't really understand that well either. We know that the locus ceruleus is affected, the innominata is also affected, and there are groups of neurons. But the most pronounced group of neurons that we lose, normally we would see by the naked eye on autopsy. If you were to look at the midbrain, you were to remove all of the outer cortex. You take off the cortex. And then you were to look at just this area, the midbrain. and you were to look at the midbrain here, by eye, without any staining, you would see these two dark diagonal bands showing you lots and lots of dopamine, lots and lots of neuromelanin with the dopamine in that group of neurons. And in an age-matched control that has Parkinson's disease, you can see that most of the neuromelanin and therefore the dopamine is lost. That dark staining that you see here, that dark band on both sides, is usually lost in Parkinson's disease. The loss of these cells means that you're losing voluntary movement and the smoothness of the voluntary movement as well. Again, we don't know a lot about Parkinson's disease. We know that it is continuous. Once you start developing the signs and symptoms of Parkinson's, can any of you tell me what you think is a sign and symptom of Parkinson's, by the way? Like, what's an early sign and symptom of Parkinson's? What do you think? So obviously, outside of movement. So you might have some movement disorder or motor dysfunction. Outside of that, yes? Sorry? Tremor. You might have tremor. Yes, you might have unusual spasms. Anything else not related to movement? Yes. Speech, so oftentimes like problems with motor coordination around swallowing speech, that also accompanies things like Parkinson's. But even before that, like you'd want to know, before I start developing these tremors, before I start not being able to move properly, before I can't turn normally, which is often a big case with Parkinson's, what happens to these individuals? Other things. Yes? Could be memory loss. The most often reported things, and we'll talk a little bit about the gut microbiome, but outside of the gut microbiome changes, the sort of changes in the bowel movements, are also things like sleep disturbances. So often sleep disturbances are one of the early signs, and if you happen to talk to a neurologist over at Toronto Western, they'll tell you that one of the first questions that they ask for individuals that they suspect have Parkinson's, do you have problems sleeping at night? Does your partner notice that you have problems sleeping at night? So there are a lot of symptomatic things that people look for now that, again, as neurobiology students, I'm hoping that you'll think about in this context. We have no known cure for this. We know that there are causes for Parkinson's outside of genetics that we can account for. So that 90%, one of the questions that you might be asked is, do you have any history of having atherosclerosis in the cerebral cortex? Have you ever had a very, very severe form of viral encephalitis? Because if you had that and you develop Parkinson's, that can actually be traced back to the viral encephalopathy that resulted in Parkinson's disease. Have you been taking antipsychotic medication? So have you been taking things like reserpine or other drugs that can actually impact the dopaminergic system? Then that can account for Parkinson's disease. But outside of all of these questions, we really don't have a root cause for Parkinson's disease. So again, before I go on to the next slide, let me ask you a question. Which of these diseases or disorders do you think we actually can describe better, or we know more about? Do we know more about the history, the long-term longevity and the history, in recorded history, of depression or Parkinson's disease? Which one do we know more about for a longer period of time? Parkinson's? Any other guesses? Depression? Any other guesses? No, you either have to guess Parkinson's or depression, right? So the short answer is that we actually can go back in recorded history and find very clear evidence, even at the time of Hippocrates, of things like depression. And people tried to understand depression way back then, but Parkinson's disease actually in the scientific literature, in any form of literature, actually didn't exist until the 1800s. So this becomes a really interesting question. If it's not reported, did it actually exist before the 1800s? How many of you say yes? Parkinson's disease has been with us since the dawn of time. Some of you? Okay. How many of you think it's relatively new, after the 1800s? Okay, and again, it's about 50-50. The answer is we may never know. And there are some pieces of evidence that I'm going to suggest to you today that might suggest that it's actually a modern disease that, and that might be one of the reasons why we don't know much about it. So again, I don't need you to memorize this. I think that is in your pre-lecture checklist. If you've read through your pre-lecture checklist, you don't need to know all of these different forms of features that are associated with Parkinson's disease. I'm just going to mention rigidity, because rigidity actually becomes important in the story that I'm going to be telling you in a few moments. Have any of you heard of wax-like rigidity or wax-like flexibility? So those of you who might have been in a neurology clinic, can you guess what that might mean? So the rigidity part you all get, like something that's really stiff, you can't really move it well. Like what's wax-like rigidity or flexibility? Yes? Okay, that's a really good explanation. I'm going to add just a little bit to it. And this rigidity is important. So if I put your arm up, I'm a neurologist, I put your arm up like this, and you hold it like this, it will actually go down very, very slowly. Kind of like melting wax, but it goes down very, very slowly. And this wax-like rigidity and flexibility becomes an important story in a few moments. So we're actually really interested in the opposite side of things. We want to know, is there actually a link between the gastrointestinal system, the gut microbiome, brain inflammation, and Parkinson's disease? That's what we started out with. That's that Cell paper that I showed you a little bit earlier on. So again, a number of these different disorders have actually been fairly well described. And one of the things that is becoming more and more evident, when you're looking at the CNS, the brain and the spinal cord, the characteristic features of the CNS, it's now becoming more evident that the periphery actually plays a big role in the development of neurological or neurodegenerative conditions. And again, you can go back and if you're looking at the gut microbiome, John Kyan's lab in Ireland is actually one of the best labs that have been looking at this for the longest period of time. Again, this is fairly new too. So it used to be thought, now that people bought into the gut microbiome, that yes, what happens in your gut actually can impact what happens up in your brain. So I'm going to try to convince you of that in today's lecture and again in the next lecture. But it also works the other way. This is that bidirectionality. This is actually fairly new. This is about 5 years old, where people now think that your brain can actually impact your gut. And again, it may sound like it makes perfect sense. Why wouldn't your brain impact the gut? When I'm stressed out, I don't eat the same kinds of foods. And if I'm chronically stressed, I don't normally eat the types of foods I normally would eat, etc. And if I'm taking medication to treat the stress, etc., all of that will impact the gut. So it's now thought to be bidirectional. It's not just the gut affecting the brain now also affects the gut, and we're also really concerned about this bidirectionality. This becomes important when we get into the modules related to mental health. When we talk about anxiety and we talk about depression, we know that nociception, the sensation of pain, is also very heavily impacted by the gut microbiome, and we now have some ideas about how nociception is affected by this. And there's quite a few papers now that have come out suggesting that changes in the gut microbiome might be impacting autism spectrum disorders as well. So here are a couple of things, and the next couple of bullet points are actually somewhat important for you to make sure that you know. So we know that the GI physiology, so all of you have taken some kind of physiology course, the GI physiology and the way that the GI tract actually moves, the way that it contracts, are very heavily influenced by the signals that are coming both from the enteric nervous system, the nervous system that controls the gut, as well as from the CNS. Again, a couple of other things to make sure that you are all aware of. It's one of those trivial facts that most people now know, but I wanted to make sure that you understood that there was a scientific basis for this as well. The gut is actually the biggest source of neurotransmitters. You think that your brain makes a lot of neurotransmitters, it does, but in fact the greatest source of neurotransmitters is actually the gut. Serotonin, for example, is produced at higher levels within the gut than it is within, for example, the brain. So when we're looking at the effect of different drugs, oftentimes we look at the effect of serotonin inhibitors on the gut as well. So again, we know that neurotransmitters, all of the different neurotransmitters that are found in the brain are produced by the gut. We know that immune signaling is also very big within the gut, and we'll be going through this. hormones, neuropeptides, all of these things are impacting the gut and therefore also impacting the brain. We just don't know all the mechanisms. This is somewhat new territory for us. We've been looking at this since roughly the year 2002, 2003, but we really are just beginning sort of this process of examining this. So again, there's a lot of research that's been developing recently to look at how the gut microbiome can affect neurodevelopment. There's some strong evidence to suggest that autism spectrum could be impacted by neuroinflammatory markers and the immune system responding to changes in the gut microbiome. Again, fairly new, still somewhat controversial, and we'll have to wait to see if that story actually comes out. Now, I wanted to mention a couple of things. In these studies that I'm going to be showing to you, and it's a very important point, for those of you who have studied MGY or other courses, I want to make sure that we're all on the same page. We have two different types of mice. We have mice that are completely germ-free. They have no microbes ever in their life. From the time that they're born until the time they die, they have like no microbes at all. And those are germ-free mice, completely germ-free. They are born and bred in a sterile environment, and they have no, none of these different bacterial exposures, etc. These are different than specific pathogen-free or SPF mice. These are housed in containers that will limit the types of bacteria, but overall it's not completely germ-free. The other thing to make note of, even though they're not completely germ-free, therefore they have a gut microbiome. SPF mice have bacteria living within their gut. We can actually wipe out every last one of those bacteria by using high doses of antibiotics. So we can actually do antibiotic-treated SPF mice, which are the two different groups we normally look at. This would be the control, no gut microbiome versus a gut microbiome that we can then completely eliminate through antibiotics. One of the things that we know now is that the gut microbiome can actually play a role in hippocampal neurogenesis, the generation of new neurons. And we'll come back to hippocampal neurogenesis and anxiety. And this can lead to impairments in spatial recognition, object recognition. So the gut microbiome can developmentally regulate how the hippocampus is forming and neurogenesis within the hippocampus, according to a few studies. We know that the gut microbiome can also regulate the expression of a number of serotonin receptors. The 5-HT1 serotonin receptor, we know that it can impact brain-derived neurotrophic factor, or BDNF. It can also impact the levels of NMDA receptor subunits. So overall, the gut microbiome looks like it can affect things that are going on within the brain. It can affect receptor levels, it can affect BDNF. BDNF can affect the overall health of neurons. All of this suggests that the gut microbiome actually plays a heavy role in impacting the brain. So again, the microbiome also increases the amount of circulating serotonin within your body, at least in mice, also now shown to be true in humans. It will therefore affect anxiety levels, hyperactivity, as well as cognition. So your gut is producing neurotransmitters. It's also producing factors that can directly impact your brain. So again, I'm going to switch gears back just a little bit. Right? And before we get into the paper, I'm going to talk a little bit more about that question that I posed to you earlier. Is Parkinson's disease an industrial disorder? We don't know. Like, our best guess is that it's probably something that is in the environment, because we can't understand the genetics. And as a result of not understanding the genetics, we're looking for other causes. And most often, this industrial disorder is sort of brought up as a countervailing hypothesis. So is there a cause? So are you sitting in a room right now, because this is a theory, are you sitting in a room right now where the walls have like black mold, and the black mold is producing this volatile ethanol? You breathe it in. The more that you're sitting here being exposed to it, the more likely that you will develop Parkinson's disease. Is there an environmental cause? We don't have a lot of genetic causes, so this makes some sense. If we were to take a look at the overall population health, when we look at the incidence of Parkinson's disease, for example, one of the things that most often comes up is that when you look in rural communities, communities where there's lots of farming and agrarian farming, and they're using lots of pesticides and insecticides to control their crops, then one of the things that comes up is in those areas where there's lots of pesticide use and lots of insecticide use, there's an increase in the overall prevalence of Parkinson's disease. So is it just correlational? Is it causational? Again, we have no idea which one is being the cause, or if this is just sort of happening as a result of a number of other factors. We again have no idea. So is it something that is virally mediated? There's a lot of evidence to suggest that viruses can somehow also kill dopaminergic neurons in very specific ways, and we'll talk about that in a different context later on. But this gets into this really interesting hypothesis. This interesting hypothesis is this: that something in the environment can actually be taken up into your brain, it's taken up and then kills your dopaminergic neurons. So do we have any evidence of this? We have one piece of very strong evidence from 1982 and 1983 that suggests something in the environment actually kills dopaminergic neurons. And this is sort of the evidence that we have that it is not genetic, that it is environmental. Because this drug, MPTP, is actually a good sort of indicator that it could be a chemical that is being released that somehow causes dopaminergic neurons to die. How many of you have heard of MPTP? Oh, okay, William, how do you know MPTP? Okay, so it's a drug. How many of you have used MPTP in research? Have any of you used MPTP at all? Have you heard of MPTP? No? Okay, so I'm going to take just a little, I'm going to take 2 minutes, and I'm going to give you a really strong warning if you ever decide to work in a lab with MPTP. So I was not a really good student, as many of you will hear several times, and I'm sure that you realize that it's carried over into being a prof too, but when I was an undergrad student, I wasn't really a great student. I actually got into a lab almost by accident in grad school. I applied really late, I didn't get into the labs that I wanted because my big hope was that I would get into a physiology lab that dealt with cardiovascular or renal systems, because renal system physiology makes perfect sense, so does cardiovascular physiology, the nervous system, and these stupid jumping action potentials over these nodes of Ranvier. I have no idea what they were talking about. So it was my last choice, but I got into a neurophysiology lab, and because I was so desperate to be in graduate school, when I joined the lab, the very first day, the PI came up and asked me, have you ever worked with animals before? And because I was in undergrad, I had no undergraduate research experience, and because I really wanted to be in his research lab, so I didn't want to be sitting at home doing nothing, I lied and I said, yes, I've worked with like animals all my life. I can do anything that you ask. And so he says, okay, great, I have this project for you that you're going to be working on with my postdoc, and you're going to be inducing Parkinson's disease using this compound MPTP. And I said, I can do this. And so the next day I showed up bright and early. I sort of done some research on MPTP, and I read a couple of papers. So I showed up to the lab, and the postdoc that I was assigned to took me into the lab next to ours. I'd never been in there because it was like day 2 of my lab experience. So I'd read about MPTP. I'd worked in sort of a 3rd-year undergraduate lab, so I knew how to weigh out chemicals and everything else. But now I'm kind of realizing that maybe I shouldn't have lied about working with animals, because they take me over to this special fume hood, and this fume hood says biohazard, like all over the place. And then they show me like the special gear that I'm supposed to be wearing, and I put into like one lab coat, then I put another lab coat on top of that lab coat, then they're starting to take down my lab coat, and then they put my gloves on, and they put another set of gloves on, and they tape off my gloves, and then they say, okay, inside that fume hood, you're going to find this special can opener that you're going to use to open up that gold canister of MPTP. So my hands are kind of shaking by this point, and I'm kind of like trying to act really cool, and I'm trying to talk up the postdoc, and I'm opening up this vial of MPTP. And eventually, somehow, I magically got it open, and after opening up this canister, there's another canister inside. You have to open that one too. So again, on the outside it says, biohazard, do not take, causes Parkinson's disease. So now my hands are like going nuts, right? So I can barely control them. So I'm taking this and kind of trying to chat up the postdoc. There are little beads of sweat kind of going down my side of my neck. And I'm opening up this second canister. Inside the second canister, I'm praying that there isn't a third canister because I don't think I can make it. So this last canister actually has this little ampule that says MPTP. I'm supposed to break this ampule and then put it into alcohol, which dissolves the MPTP. And my postdoc has been staring at me all this time. I didn't realize this at the time, but the postdoc is actually my supervisor's wife that I didn't know, and she would eventually report back to my PI, but I didn't know this at the time. So she's smiling at me, going, "You've never done this before, have you?" I said, "Is it okay if I said that I've never done this before?" And she says, "Yeah, it's probably a good idea to tell me that now before we start injecting." So the bottom line is, always be honest. If you're going to join a research lab, don't work with MPTP. It's a really scary substance. And one of the reasons why it's so scary is because, as William pointed out, in the 1980s, there were a number of cases where different people were creating synthetic heroin. And the synthetic form of heroin that you see here, on the left, on the bottom, was actually what people were trying to synthesize. And there are many famous cases where people try to synthesize synthetic heroin. But one of the issues is that if you don't keep clean glassware, then over time, this synthetic heroin actually builds up this compound here. And this is MPTP. This is the substance that actually caused my hands to shake, not because of Parkinson's, but because I was so nervous. And MPTP actually will be converted very quickly into MPP+, and MPP+ looks a lot to the neuron like dopamine. This is what dopamine structure is. This is MPP+, and it kind of looks like dopamine. That's why it targets dopaminergic cells. It doesn't really damage other neurotransmitter systems. When you inject it, it destroys fairly selectively these dopaminergic terminals. Again, this brilliant individual who was brand new director of the Santa Clara Valley Medical Hospital. This is James Langston over on the left, and James Langston sees the very first patient that has taken an overdose of MPTP. They shot up with heroin, but there was also enough MPTP that this young individual, very young, in their 20s, actually had full-blown Parkinson's. But here's the thing. When you're in a hospital, you can't tell the difference between a movement disorder and a neurological condition. If you read his original manuscript back in 1982, there was this argument between the neurologists and the psychiatrists. The neurologists were all saying, this person obviously has Parkinson's disease. They can't move. And the psychologists were saying, it's impossible, or psychiatrists were saying, it's impossible. The person's only 20 years old. This must be a form of psychosis, and that they are exhibiting different signs related to something completely different. Eventually, James Langston finds enough of these patients. He's able to do a full neurological workup. He uses that rigidity, the wax-like rigidity, to prove to all the residents that, in fact, this has to be an issue with movement disorders. It has to be a form of Parkinson's disease. He's the first to actually show that something in the environment can specifically target dopaminergic neurons. And this is why now, in animal model studies, we actually use things that look a lot like dopamine. So we can use analogs like 6-hydroxydopamine. 6-hydroxydopamine, when we inject it into the substantia nigra, kills dopaminergic neurons. Things like MPTP are analogs of dopamine. Reserpine is an MAO inhibitor, and again, found in the dopaminergic neurons as well. Those details not being so important. Let me mention one other thing to you as well. If you were to take a look at all of these different substances, the vast majority of these will actually work on one organelle. Can you guess what one organelle they all work on? Yes? So these are all mitochondrial inhibitors. They kill mitochondria. The mitochondria are attacked, the mitochondria are destroyed, and that in turn destroys the neurons that they're associated with. So we only have 2 known genetic causes, Parkin and alpha-synuclein, and I'm just going to remind you of that before we get into this paper. So again, there's something in the environment, we don't understand all of it, and some of it could be related to an environmental toxin, but people are still not satisfied with this either, because to be honest, for this to be true, all of you are pretty much exposed to the same environment all the time. So that by itself can't possibly be the only root cause. So either there has to be genetics and environmental factors, or something else plus those other 2. Do you have a question or comment?
This is kind of like, it's a really great question. What if like, you have someone who is completely like isolated from like all types of stimuli that their dopamine receptor neurons automatically like they generate somehow? Like, I don't know if that's possible, but could they essentially get all these like movement disorders from like this lack of stimulation all their life?
So if there wasn't appropriate stimulation to like groups of neurons, would that cause degeneration in and of itself? I think that's your question?
Sort of, yeah, and then would it lead to, say, these neurons?
Yeah, so I can sort of answer that for you. There are different conditions where individuals can't move. They don't get that stimulation directly. And again, there are 2 different states. One is called locked-in syndrome, and the other one is called a vegetative state. In those individuals, as far as we can tell, there are no changes to the dopaminergic terminals, even though they are locked in somewhat permanently in some cases, or if they're in the vegetative state for prolonged periods of time, like months to like years. I don't know if that answers your question completely, but as far as we can tell, the lack of activity to stimulate the dopaminergic neurons may not be the thing that causes them to degenerate. Okay, so again, a couple of other things here as well. If you go back, and this is actually a really good review, if you're interested in all of this material, if you go back far enough, people have actually looked at the microbiome of people with Alzheimer's disease, of Parkinson's disease, of different types of neurological conditions, neurodegenerative conditions. Perhaps not surprisingly, their gut microbiomes are different than the normal average individual. So that's actually been known for a while, summarized there in 2016. So if you were to take a look at the fecal and mucosal-associated gut microbiome, or microbiota, for example, in Parkinson's versus healthy controls, this has been well established. It's actually quite well established for a number of different labs over a long period of time. But this, we don't really know. Is it the cause or the effect? We don't really make a clear association this way. So we also know that there are a couple of other factors that make this unique for Parkinson's disease. So if you look at an individual with Parkinson's disease, oftentimes people that have Parkinson's disease also complain about problems with digestion. So they have gut inflammation. They have inflammation that you would not normally suspect. So it should be like substantia nigra. I get that something's breaking down the neurons within your brain, but why do they also have inflammation in their gut? And a lot of other gut abnormalities. So one of the first signs, as I mentioned to you before, is a sleep disturbance. But another sign that often accompanies and precedes Parkinson's disease is this constipation, sometimes by many, many years. And this actually led to what is known as Brack's hypothesis. Brack was one of the first people to look at the gut microbiome in the early 2000s, as I mentioned to you before. And Brack's hypothesis actually suggests, and it was a bizarre theory, I don't know if it's correct to be quite honest, but it's an interesting hypothesis. Brack's hypothesis is that somehow alpha-synuclein actually begins to accumulate in the gut first. And just like a prion disease, have you heard of prion disorders? Yes? So prions are these misfolded proteins, and they can cause other proteins to misfold when they start acting as the template for them. That somehow something in the gut goes wrong. This alpha-synuclein in the gut, because it's also found there too, by the way, it also causes it to misfold, and this misfolded protein will actually propagate or move up the vagus nerve up into the brain, because that is the one route that is a direct route from the CNS down to the periphery. That's the vagus nerve. That's the biggest route of entry. So it's entirely possible that this might actually happen. And that was his hypothesis, that the vagus nerve is the route of entry for a lot of these different neurodegenerative conditions, especially things like alpha-synuclein. So alpha-synuclein misfolding, vagus nerve propagation, I want you to make sure that you know Brack's hypothesis. So again, we also know that alpha-synuclein is found not just in the brain, we also found alpha-synuclein and misfolded alpha-synuclein in the enteric nervous system, the nervous system that supplies the gut. We find it inside the vagus nerve, we find it in the glossopharyngeal nerve, and this is also really, really interesting. If you were to cut the vagus nerve, you do a vagotomy. You don't actually need to have vagus nerve stimulation to the gut. If you do a vagotomy, individuals that would normally develop Parkinson's disease are actually at reduced risk. So again, very surprising results, suggesting that maybe there is a connection between the gut microbiome, the periphery, and somehow something goes wrong, and this gets transmitted up the vagus nerve up into the brain, at least according to Brack's hypothesis. But now we actually have to prove this. We need to be able to show, do gut bacteria regulate all of these hallmark motor deficits? Something has to happen to movement. Parkinson's is a movement disorder. If we alter the gut microbiome, can we show changes in movement? It has to involve alpha-synuclein. We have to use alpha-synuclein somehow in this. We also get the neuroinflammation related to this alpha-synuclein pathology, and we get all of the motor features that I've been trying to convince you of. And most importantly, can we actually show a direct cause between the gut microbiome and transplant that into a normal mouse and have it develop Parkinson's disease by developing this alpha-synucleopathy? So all of these things are going to be addressed in this paper. I'm going to be taking you through different aspects of it. I need all of you to make sure that you're okay with some of the molecular biological terms that I'm going to use. So I want all of us to be on the same page together. In this particular paper, they are using a transgenic mouse. So their transgenic mouse that they're using in all their studies, this is their background. All of these mice are identical in the sense that they have a Thy1 promoter, and this Thy1 promoter actually stands for thymocyte. That's where the thigh comes from. So the Thy1 is actually from thymocyte. It was a protein, a glycoprotein that was isolated first from immune cells, but we also find it in neurons. And this is why we're using it. Within the brain, Thy1 is found within mature neurons. Now, can you tell me a reason why is it important that we're looking at mature neurons? Yes? Exactly. So this is a disorder normally associated with an older brain, so it makes sense that we want mature neurons and a protein that is going to be expressed only in mature neurons. And Thy1 is normally a promoter that would drive the expression of proteins in a mature neuron. Okay, so not a young neuron wouldn't make sense or a good model for Parkinson's, but an older group of neurons. And one of the things that this is going to drive is alpha-synuclein. So remember, alpha-synuclein is the normal protein. We accumulate it, it gets misfolded. So neurons, mature neurons, are also going to express the alpha-synuclein and overexpress this alpha-synuclein. This is going to be abbreviated as ASO for alpha-synuclein overexpressing mice. And one of the things that happens is that these mice, in another study, not in this one, if you overexpress alpha-synuclein, given enough time, in those mature neurons, they develop Parkinson's disease. They stop moving as well, they lose a lot of their reflexes, etc. And these defects in coordinated motor tasks become evident about 12 weeks of age. How long do mice normally live, by the way? So how old is that? So this is the beginning of the degeneration. How old is like a mouse normally? The oldest mouse on record. Any ideas? 1 year. Okay, good guess. Peace sign, 2 years. Any other guesses? So mice usually in a laboratory setting live for about 2 years-ish, so somewhere between like 1.5 to 2 years. So this is not quite old age, so they're beginning at about 12 weeks of age, somewhere between 3 to 4 months. That's the beginning of that. So they're not quite at middle age yet, but it's still a really good model. Is there anything about this that you need to ask me about now or are unclear about? No? Okay, so I'm not going to go through all of this slide. I'm only going to go through these 4 panels, and then I'll go through the last slide related to you trying to find a paper. So one of the things that is really important, how do you assess motor changes in mice? Like, what do you look for? I can tell you from first-hand experience, having done that project where I injected MPTP into mice, what do you think happened to those mice? I injected them with 30 milligrams per kilogram of MPTP. I did that for 5 days in a row. That's 150 milligrams per kilogram total. That killed off all of their substantia nigra neurons. What do you think happened to the mice? How did I know it was working? Yes? They got Parkinson's. What does Parkinson's look like in a mouse? Tremor. Okay, there is a little bit of shaking. That's right. Did they stop moving? Mice can still move. We don't get all the signs. We don't get the slowness of movement. They shuffle a little bit more. So this is important. Understanding, like, how do we see the same phenotype in a mouse versus a human? We can't completely eliminate all of their motor activity. That would be both unethical and it would be really harmful to the animal. They wouldn't be able to feed themselves. But we can actually look at tests of motor function. So this is how we look at whether a mouse has an impairment in motor activity and therefore has Parkinson's-like phenotype. One of these is how long does it take for it to cross a beam? Beam traversal. If it takes you longer, do you have Parkinson's? Or if you cross it faster, do you more likely have Parkinson's? So if I'm taking more time to go across the beam, do I have Parkinson's? Probably. If I'm taking longer periods of time, this beam traversal becoming longer means that I'm probably slowing down. Bradykinesia, loss of regulated movement. The other thing that you can do is nasal adhesive removal. What do you think that is? Any guesses? Yes? Yeah, exactly. So as your colleague pointed out, if you put a little piece of adhesive on the nose of a mouse, you want to see how long does it take for it to try to get it off. The longer it takes, the more likely it has signs that look like Parkinson's. The other thing that you want to look at is how long does it take for it to have a reflex. So the hind limb, if you give it something to hold on to, how long does it take for it to clasp and grab the pole or the object that you are giving to it? The longer it takes to have this reflex, the worse off that it is. And then how long does it take? Because it actually requires a lot of motor movement. If you put it on the top of a pole and it wants to come down, again, the slowness of movement tells you about Parkinson's. So if we're looking at all of these different mice, and we talk about these specific pathogen-free or SPF mice, all of these, again, are abbreviated as either wild type or the alpha-synuclein overexpressing. What you see is when you have alpha-synuclein overexpression, they're always moving slower. So whether it is, again, time to get across a beam, it takes them longer to cross the beam. This alpha-synuclein overexpression, when they have a full gut microbiome, actually takes them longer to cross that particular beam. If it is the nasal adhesive removal, and you put a piece of adhesive on their nose, and you look at alpha-synuclein overexpression, if they have a complete gut microbiome, then it's also going to take them significantly longer to remove this as well. All these other indices as well, the time to descend a pole also taking longer if you have alpha-synuclein overexpression and a full gut microbiome, and the hind limb reflex, all of these are taking longer, not unexpectedly. The surprise is that if you were to look at these mice and you were to wipe out their gut microbiome, which is what the germ-free mice are, they're the same age alpha-synuclein overexpressors, we can see that the slowness in these germ-free mice, so we're looking at the ASOs in both the germ-free here and the serum pathogen-free, you can see that that delay in time is always going down. somehow by wiping out the gut microbiome through high levels of antibiotics, we can actually reverse some of the motor delays that we were seeing earlier. Does that sound somewhat magical to you? Yes? No? Not sure. So we're going to come back and talk about this in some more detail next week. I want you to think a little bit about this. I want you to be sure that you understand things related to Phi1, where it's normally found, why we would use the Phi1 promoter to drive expression of a protein like alpha-synuclein, and then we'll come back and finish off the rest of the paper. So I promised I'd let you out at about 7, so I'm just a little bit behind. I do want you to start on this early. You can bring your papers in at any time. I'll be happy to go through them with you. I'd be happy to give you suggestions on them. But I want it to be a paper that you feel comfortable with, that you want to actually explore and learn some more about. I'm actually going to try to do this myself. I'm going to do this with around the same time as all of you are doing it. And so you'll see like my progress as well. Darren, are you going to mark mine as well? Okay, we'll see. I'll kind of sneak one in and see if I get a good mark on this assignment, just like all of you. But here's one of the things that I'd like you to make sure that you are aware of. If you're going to pick a paper, try to avoid picking a paper that already has a visual graphic or a visual summary associated with it. You'll know as soon as you open up the paper, there's a little 3-panel figure. Please try to avoid that. You can still pick it, but you'll have to come up with your own unique one. So that becomes somewhat of a challenge if it already is out there. I'd like you to pick a recent paper, something published between 2012 and 2017. So one of the reasons why I'm asking you to pick something recent, science is changing so quickly that we know that papers that were published before 2011 sometimes have been overturned, and we know that they're not quite as accurate as they could be. I'd like you to also make sure that it interests you. It could be any field. It could be psychology related. It could be cognitive science related. It could be something that is artificial intelligence related that I'm not going to cover in this course, but it's really interesting to you that you want to share with a broader audience. I want you to be able to create the same sort of 3 to 4 panel abstract or graphic abstract that I showed you earlier today. It's going to be due by October 18th. I'm going to check in with you frequently to make sure that you're on track. You have lots of time, but you're going to find that the hardest part is to actually find the paper that you're going to be sticking with past the graphic abstract, etc. Again, if you bring the paper, I'll be happy to tell you, yes, this is a great paper. Please go ahead and do it. Here's some suggestions for you. My job is to make sure that all of you in this class can get that 5 out of 5. And that's my goal, that you'll all get a perfect mark on this particular assignment. Do you have any questions on this before we wrap things up? No? Okay, so again, please make sure you're contacting me if you have any questions, and I'm hoping that you have a great rest of the week. I'll see you all back here next Wednesday.